Hello and welcome back once again on the Biobasics. This is Dr. Komalacharya and this time I'm going to talk about gene regulation and tryptophan operon. We have seen in our previous video lectures that gene regulation falls into categories: regulation of gene expression and post-translational regulations. Further, the gene expression regulations can occur at two different levels. One is at transcription level and second one is at translation level. In last video lecture, we have seen that in lactose operon, the transcription of genes is regulated at initiation step. But apart from that, transcription can also be controlled at elongation and termination steps. In such regulations, transcription begins but stops prematurely. One example of such operon system is tryptophan operon. The tryptophan operon of E. coli controls the synthesis of enzymes that catalyze the biosynthesis of amino acid tryptophan. There are 5 different structural genes that encode for different enzymes which convert chorismic acid to tryptophan, whose expression is regulated by tryptophan operon. Now, as far as the structure of the tryptophan operon is concerned, there are 5 different structural genes: TRPE, D, C, B, and A. Each encodes for different enzymes responsible for the conversion of chorismic acid into tryptophan amino acid. Apart from that, there is a promoter region, there is an operator region which is overlapped with the promoter region, there is a ladder region which is composed of ladder sequence and attenuator sequence, and apart from that, there is a regulatory gene TRPR which is distantly located from the tryptophan operon but encodes for tryptophan repressor protein. So this was the structure of the tryptophan operon. Now let's see how it works. So the expression of tryptophan operon is regulated by two different mechanisms. One is repression, second one is attenuation. So let's see the repression first. Now the tryptophan operon is negatively repressible operon system, which means that if the tryptophan is present, then this operon system will be turned off because it does not require the enzymes needed for the tryptophan biosynthesis. But if the tryptophan is not available, then it will require the enzymes needed for its biosynthesis. So the operon will be turned on. This turning on and off is occurred due to the repressible protein. There is a regulatory gene TRPR which encodes for a repressor protein, that is TRPR protein. This is a homodimer which is having two subunits. Each subunit is made up of helix turn helix motifs like this. This can bind to the operator region of the tryptophan operon, but this cannot bind on its own. It requires the activation by a co-repressor, and here the tryptophan itself serves as the co-repressor. It binds to the inactive form of the repressor protein, convert it into its active form due to some conformational changes into it, and then this complex co-repressor with the repressor protein will bind to the operator region of this tryptophan operon. Now how this system works? So there are two different cases. One is in the presence of tryptophan. So if the tryptophan is present, then it will serve as the co-repressor. It will bind to this inactive form of repressor, convert it into active form. Now this complex will bind to the operator region of this operon. And when this RNA polymerase will bind with the promoter region, it will try to carry out the transcription of these structural genes. But this repressor protein will block its path towards this structural genes. So it cannot transcribe the structural genes of tryptophan operon. And if the tryptophan is absent, then what will happen? Now the tryptophan is absent, so it cannot bind to the repressor protein. So inactive repressor cannot convert into the active form, and so it cannot bind to the operator region. So when the RNA polymerase will bind to the promoter region, it will carry out the transcription of the structural genes, and so different enzymes responsible for the tryptophan biosynthesis will be produced. So this is the way the tryptophan operon is regulated at initiation step of transcription through repression mechanism. But what if the transcription still continues by RNA polymerase? Then it will be controlled by another fine mechanism, that is attenuation. So When the RNA polymerase will begin the transcription, then it will reach the ladder region present between the operator region and the first structural gene. Then the transcription is terminated within this region, ladder region, and this is called attenuation. Now this attenuation becomes possible due to the fact that in a bacterial cell, transcription is coupled with the translation process. Look at here. This is the DNA from which the RNA polymerase is synthesizing the mRNA strands through transcription process. And as the mRNA, nascent mRNA is synthesizing, the ribosome will bind to it and it will start producing or synthesizing the polypeptide chain from it. So this is the way transcription and translation, both the processes are going on simultaneously. So this is the main base why the attenuation process becomes possible. There is another important thing. Attenuation also becomes possible because of the presence of nucleotide sequence present in the ladder region. This sequence is known as attenuator. Now what is this attenuator basically? It is a row independent termination site which is rich in GC rich segment followed by the poly U sequence. Now let's take a brief review about this row independent termination site. In bacteria, basically there are two different strategies for transcription termination. One is row dependent termination and second one is row independent termination. As far as row independent termination is concerned, it involves an intrinsic terminator which has GC rich inverted repeats. So the RNA transcript formed after the transcription is having abundant GC base pairs, which results in the formation of hairpin loop-like structures followed by this poly U sequence. Now, due to this poly U sequence, the RNA polymerase will pause over here for some time. Besides that, the AU base pair over here in DNA RNA hybrid is too weak to hold this hybrid structure. So the RNA will be detached or will be released from here, followed by the dissociation of RNA polymerase from here, and thus the transcription will get terminated. This kind of row independent transcription termination site known as attenuator is present in the ladder region. Now let's take a view of ladder region. The ladder region is located between operator and the first structural gene TRPE. That is called ladder region. This ladder region controls the continuation of transcription of structural genes even after the RNA polymer is bound to the promoter region. Okay, now this ladder region is composed of two things, ladder sequence and the attenuator sequence. This ladder sequence is the first sequence to be transcribed by the RNA polymerase. But the striking feature of this ladder sequence is that it is divided in four different stretches. 1, 2, 3 and 4. All of these stretches are made up of complementary base sequences, so they can make base pair with each other and form hairpin loop-like structure. Basically, this ladder sequence has 14 codon open reading frame that codes for the ladder peptide. These codons include 2 codon for tryptophan at position number 10 and 11. There is also a stop codon located in the gap between segment number 1 and 2. Now, due to these complementary sequences, there are 3 possibilities. 1. Segment 1 and 2 make base pair and form a hairpin structure. Segment 2 and 3 make a base pair and form a hairpin loop-like structure like this. And segment number 3 and 4 can make a base pair and form a hairpin loop-like structure like this. The most important part of this system is that this 3-4 hairpin structure, hairpin loop, contains the attenuator sequence because it is followed by the poly U sequence also and form a transcription terminator site. Now let's understand this thing thoroughly. See, this is the tryptophan codon. This is segment number 1, 2, 3 and 4. So if the 1 and 2 segments will form a loop-like structure, then segment number 3 and 4 will bind to each other and form a hairpin loop structure followed by the poly U sequence. If this thing occurs, then this is the row independent transcription termination site and so the transcription will be discontinued. Now, if the segment number 1 is not making base pair with segment number 2, then segment 2 will form a hairpin loop like structure with segment number 3, and thus the segment number 4 will remain free, followed by the poly-U sequence. And so the row independent termination site is not forming over here, and thus the transcription will be continued. Now I have told you that transcription and translation is going on simultaneously in bacterial cell. So we have to understand the translation first. So let's take a brief review of it. This is the mRNA structure, this is the ribosome, this is the charged tRNA, and this is the synthesizing polypeptide chain. So as the nascent mRNA strand has been synthesized due to transcription process from the DNA, the ribosome will attach to it and it will start synthesizing the polypeptide chain from it, and all these processes are going on simultaneously. So now let's understand this mechanism very deeply. If the RNA polymerase escapes the repression, then it begins the transcription until it reaches the pause site located between segment number 1 and 2 of the leader sequence. Simultaneously, the ribosome will begin translation of the leader mRNA, and when the ribosome reaches the paused RNA polymerase, the RNA polymerase will resume its transcription. This pause serves the important role in synchronizing transcription and translation process. Now further fate of this process will depend on the concentration of tryptophan. If the tryptophan concentration is low, then what will happen? When the ribosome will reach to the tryptophan codon present in the segment 1, it will not have the tryptophanyl tRNA to bind to it, because there is no tryptophan available for it. So the ribosome will halt over here and it will capture the segment number 1. Look at here. So now it cannot bind to the segment number 2. So now 2 and 3 will make a hairpin loop like structure, and the segment number 4 will remain free. So here the transcription termination site is not created. So the transcription of the tryptophan operon will be continued. Now if the tryptophan is available in abundant amount, then what will happen? If the ribosome will reach the tryptophan codon, then there will be enough tryptophanyl tRNA for it. So the ribosome will carry out the translation process, and this is the stop codon. So when the ribosome will reach over here, the nascent polypeptide chain will be released from here, and now the segment number 1 will bind to the segment number 2. So the 3 will be available to make a hairpin loop like structure with segment number 4, followed by the poly-U sequence. So now this is the transcription termination site, and so the RNA polymerase will stop transcribing the structural genes, and so the enzymes responsible for the tryptophan biosynthesis will not be produced. So this is how the tryptophan operon system works. If you like the video, please click the like button, please share this video and follow for more such video lectures. Thank you very much.